Hello and welcome to Unsupervised Thinking, a podcast on neuroscience, artificial intelligence, and science more broadly. We are a group of neuroscientists. I'm Grace.
I'm Josh.
And I'm Nancy.
And for this episode, we're going to be talking about glia. What are they doing? They are cells in the brain, but they're not neurons, so what are they for? And so for this, we read The Mystery and Magic of Glia, a perspective on their roles in health and disease by Ben Bars, and that's a neuron perspective from 2008. And we read Glia, Listening and Talking to the Synapse by Philip Hayden, and that's a Nature Reviews Neuroscience piece from 2001. So we have Nancy with us, who's our resident experimentalist, which is nice to have when we're being reminded of the fact that the brain is a physical thing. Nancy, would you like to remind people where you are and what you do?
Yeah, sure thing. So I'm a postdoc. I know Josh and Grace from when we were all together in grad school at Columbia University, and they were my theory friends, and I was the experimentalist in the room. And now I'm a postdoc at MIT in the lab of Kay Tai, but we're actually moving to Salk Institute. So in a couple of weeks, I'll be a postdoc in the Salk Institute. So I study social behaviors and how the brain encodes social behaviors. I use mice, just in case anybody's curious.
Great, thank you. Right, so let's get started with some basic facts about glial cells. I'll also admit that in preparation for this, I read the Wikipedia page on glia because that's where I'm starting from. And I think that's where a lot of neuroscientists would be starting from, because glial cells are not the most studied thing in neuroscience. They are usually thought of as supportive cells. They help the neurons do things, and some of the functions that they're usually known for, according to Wikipedia, are holding neurons in place, supplying nutrients and oxygen, insulating neurons, and destroying pathogens and cleaning up dead neurons. So this all feels very physical and not really related to, or at least not directly related to, kind of the cognitive functions of the brain or the behavior that the brain outputs, which are normally the topic of neuroscience study. But obviously those roles are important. I think we're also going to talk about how that description of glia, the kind of traditional description of glia that most of us probably saw in textbooks, if you saw any description of them at all, how that might not be the full picture, and how in some somewhat recent years people have found roles for glial cells that are more related to computation and to what neurons are doing. So the basic types of glial cells, there's a few different types, and I should say that the name glia comes from the Greek word for glue, which also speaks to their not primary role, but their role in holding things together. So astrocytes are a big type. They are the most abundant type in the central nervous system, so in the spine and the brain. There are oligodendrocytes, which are the particular glial cells that form the protective coating around the axons of neurons. So if you've seen a drawing of a neuron that has like little white puffy things around it at various points on the axons, that comes from the oligodendrocyte. That's the myelin sheathing. And so kind of the equivalent of oligodendrocytes in the peripheral nervous system, so kind of near muscles and things, are the Schwann cells. They provide the wrapping around axons in the peripheral nervous system. And then there are also radial glial neurons that are involved in neurogenesis. So when new neurons are born, they're involved in that process and they guide them.
And then there's microglia. I don't know if you mentioned microglia.
Oh, no, I didn't. Would you like to talk about microglia?
Yeah, sure. So microglia, by 2008, we didn't know much about microglia. And even myself, like Grace mentioned, like I didn't know much about microglia, but I happen to have a colleague that is obsessed with microglia. And she's a technician and she wants to study that in grad school. So I've benefited from talking to her about microglia. So in the past 10 years, it's become more clear that microglia are cells that respond to infections, and they go around and they literally move, which is really cool. I thought it was really strange. They move around. They sense the chemicals around them and they move around and they engulf parts of the synapse to like do synaptic pruning, for example, or if there's bacteria or some sort of infection, then they can engulf that too. So they're kind of like police in the brain because they move around and they're like really paying attention to the environment. So, but the very, very important is that they help with the synaptic pruning for during neurogenesis and during development. So
elaborate what you mean by synaptic pruning. Right.
A new neuron is going to have more synapses than it needs, and then you want to like kill off and like chop off the parts that you actually don't need.
So they're like over-connecting. They're like connecting to too many cells and forming too many synapses with other cells, and then that would be bad if they kept them all.
Exactly.
Right, so yeah, the glial cells have a lot of different functions. In one of these articles, they said that they're as diverse as neuron types, and there's a lot of different types of neurons. And there was another part where it said that it doesn't necessarily make sense to call them all part of a same class just because they're so different, but they are the cells of the brain that aren't neurons, and we call them glia. And also there are different accounts of how many there are. The original picture that I always heard was that there are 10 glial cells to every neuron in the brain. It seems like maybe an updated view puts them at roughly equal to the number of neurons in the brain. I'm not sure, but also this is a number that varies across species. So species that are lower on the evolutionary tree seem to have fewer glial cells. So C. elegans, the little worm that's studied a lot in neuroscience, they have 1 glial cell for every 5 neurons, so fewer glial cells than neurons. And then in human brains, it's at least 1-to-1 and possibly more.
So perhaps that speaks to the importance of glial cells in cognition or maybe in complexity of processes.
I think it's unclear. It could also be about size, right? I mean, if the myelination is a significant role for at least a subset of glial cells, and brains get larger, you would maybe expect that you would just need more glial cells, even if they're only related to these supportive roles.
Maybe, yeah.
I'll be the cynic here.
Yeah,
no,
okay.
Just so we have someone who's skeptical.
That is convenient. It's good to have a villain. But yeah, so obviously there are people who think that these cells play a big role in things other than just pure physical support. We can get into what happens at the synapse in particular. As Nancy said, the microglial cells are involved in synaptic pruning and kind of getting rid of synapses that don't need to be there, but the astrocytes, the most common type of glial cells in the brain, they seem to be involved in kind of maintaining synapses that should be there and maybe changing their strength and regulating what's going on there. So we can talk about the detailed ways in which they do that.
Sounds good.
I think one thing that's interesting off the bat is that glial cells have receptors for neurotransmitters. It seemed kind of like for every neurotransmitter. They have receptors for glutamate, like the excitatory neurotransmitters. They have receptors for histamine and acetylcholine and GABA, which is the main inhibitory neurotransmitter. It's like if neurons are firing and releasing neurotransmitters, the glial cells will hear about it. Like they're going to know, they're going to respond to those neurotransmitters that are floating around, and they're going to do something about it.
Right, so... And it seems, at least, I'm not sure if in the 2001 review that was there, but in response to some of these, so if some of these receptors get active, then they'll have an influx of calcium. And then they can release either glutamate or ATP. So that is transmission, right? So I think that it has to be doing something very important.
Yeah, so these glial cells, they respond to neurotransmitter. They have their own internal dynamics that involve calcium, and then they do seem to release some of their own stuff as well. And I mean, we can describe neurons in a similar way. They respond to neurotransmitters. Mm-hmm. They have their own internal dynamics. The internal dynamics of a neuron usually involve electrical changes, like the membrane potential changes, and glial cells don't seem to be that kind of excitable in an electrical way. But so their internal signaling is more in terms of calcium and other things like that. But then, they have an output based on their inputs. It's just that the glial cells are working on a much slower time scale than neurons, and a different spatial scale as well, kind of.
So one thing that I found, it seems like since maybe late 1990s, these calcium glial waves have been reported. And I was like, what's happening during these calcium glial waves? And it seems like it precedes glial transmission. So these calcium waves happen before they release either ATP or glutamate.
Yeah, so maybe let's explain the mechanism underlying the calcium waves. The claim was at least that in sort of in vitro, you can mechanically stimulate glia, and they demonstrate these calcium waves, which is the release of calcium from intracellular stores, right, for the glia, and then there's some sort of potentially gap junctions connecting to other glia that
so they
induce...
can. So that means that they can activate each other through the gap junctions. That's how they can spread.
Yeah, so the mechanism that includes the gap junctions included another molecule that is responsible for releasing the stores of calcium that exist inside the glial cells. So the glial cells have like internal organelles, as all cells do, and they have calcium stored in them. And there's this molecule that can release that calcium into the cytoplasm of the glial cell. And so that's kind of what this calcium wave is. It's like seeing the calcium being released into the goo inside the cell.
The goo.
The goo. That's what cytoplasm is. And so that molecule that caused it to be released in one glial cell, in one astrocyte, can move through the gap junction. So gap junctions are just where two neurons physically connect. It's not like a normal synapse where there's a junction that chemicals have to go across. It's like the membranes of the two neurons are connected. So that molecule can walk through that gap junction and then cause the same process in the next glial cell. So make it release its internal calcium. So it's not the calcium that's going through the gap junctions, as I understood it. It's the molecule that causes the glial cell to release its own calcium.
Yeah, you're right.
But so the visualization of it as a wave is simply that you're observing calcium concentrations, possibly via fluorescence or something like this.
And
Yes.
then there's a chain effect then. That's why
There's a
they
chain
call
effect
it.
where it propagates from one glial cell to neighboring glial cells and it sort of radiates outward or something like this.
Yeah.
And they said that in vitro, the mechanism by which they study this is that they can reliably induce calcium waves by sort of mechanical stimulation of a glia.
Yeah, you can like poke the astrocyte and it will cause it to release its internal calcium stores, which can then cause other neurons to release their internal calcium stores. But that's less clear to me. It makes more sense to me when it's like via a neurotransmitter receptor, like something attaches to the glial cell, causes like a big chemical cascade, a bunch of stuff moves around, and this molecule gets produced that releases the calcium. I don't know how the mechanical stimulation causes this.
Yeah, I mean, they elided over this a little bit, but they were simply suggesting, I think, that it's like a model of physiological stimulation.
Sure, yeah. It's a way to do it in the lab to get calcium released and to start one of these waves so that you can then study the effects of these waves. I don't think that they think that in the brain it's mechanically
Yeah, no, but I mean,
released.
I guess you have to be... I did think this as I read this. This is one of those settings where I feel like you have to be pretty comfortable that the mechanism that you're using to artificially induce calcium waves resembles, like you're studying something that you don't know how it works, and you, like, in order to believe that you're doing relevant science, I think you'd have to really strongly convince yourself that, let's say, mechanical stimulation is a good proxy for physiological stimulation.
They talk a lot about how, there's findings for some of these, like the glial impact on synapses and whatever, like that it happens in cultures of cells in dishes or it happens in slices, and that there needs to be validation of that in the actual animal, in the brain as it's working. Yeah, because sometimes you don't know when you do these like very reduced preparations of the cells if they're functioning in the same way that they do normally. But actually, the calcium waves have another means of possible transmission that they talked about, which was an extracellular way, because they said that you can get calcium waves even in cells that are physically separated, as long as there's like extracellular stuff that they can communicate through. And so that was through ATP, which anyone who's taken like an intro bio class probably recognizes as like a energy molecule, like a way to store energy. So I didn't actually know that cells, or maybe I did at some point, but I definitely forgot that cells use ATP as a signal to each other. That seems kind of surprising to me, given that it's like has this crucial role in energy.
I remember ADP being part of intracellular cascades of, molecular events, but I I didn't know ATP serves for transmission of this type across cells.
And ADP is like what happens when you break down ADP?
Exactly.
ATP?
So when you use the ATP for energy, then you remove one phosphate and then you have ADP.
Yeah, so it's adenosine triphosphate turns into adenosine diphosphate.
Okay. And so that makes sense, though, because that's just like, oh, this is like the garbage of producing energy, so
Right.
let's use it as a signal. The notion that you use, like, your actual energy as a signal, it's like cultures that use, like, beans as currency. It's like, wait, but what if you actually just want to eat that? then you're out of currency now.
But it's also probably, I mean, from an evolutionary standpoint, it's probably a fairly compelling signal, right? If you're, like, trying to get another cell to pay attention to you, it's like, hey, pay attention to me. You send it a bunch of energy.
It'll really pay attention to you.
It'll be like, I want to figure out what I can do to make that cell
Send
more energy to me.
But not if the cell that receives it also has to use it as a signal and can't use it as energy. That's the weird bit.
Well, maybe
Anyways,
there's a lot of it. Maybe there's a lot of ATP around glial cells.
the brain does seem to have a lot of energy stores because the sheathing around axons is just made of like fats and stuff. So it's probably fine. But yeah, so that's another way for these calcium waves to go. The glial cells can release ATP that then activate another glial cell to have its own calcium release. And so they don't actually need to be connected via these gap junctions. But yeah, so this kind of signaling between the glial cells is a way in which they're neuron-like because they're taking in signals from each other and sending out signals. But as I said, it's just happening on a different time scale. So the thing that they say in the paper is, neurons are communicating on the time scale of meters per second, so they can send information long distances quickly, and glial cells are communicating on the time scale of micrometers per second. So they're this very slowly diffusing signal that can't be playing the same role as neurons do, but that doesn't mean that they're not playing some interesting role.
So I found a recent paper. So from this idea of you have astrocytes that can sense ATP, and then that triggers a molecule inside that then triggers intracellular calcium, and that precedes release of ATP to the environment or glutamate, and that could activate either neurons or astrocytes. So now they're using actually optogenetics to activate astrocytes. By activate, meaning cause an artificial increase in calcium, which would then trigger release of ATP or glutamate or whatever transmitter it has inside.
This is a new way of experimentally probing.
Exactly. So, from this paper that we reviewed is from 2001. So this paper that I'm talking about is from 2017. So more recently, people have shifted to activate optogenetically astrocytes in similar ways that they do with neurons.
Are they using this for calcium wave propagation or other?
They're using it while they patch neurons on the surrounding to see what's the effect on the neuron. Essentially, they're using it to study glial transmission.
Cool. Yeah, that sounds interesting.
This one particular paper showed very convincingly that if you cause glial transmission with optogenetics, you have changes in neuronal activity in the neurons around it. So, and they showed even through which receptors. So turns out, I'm also kind of surprised of thinking about ATP as a neurotransmitter, but there's a receptor called P2Y1R, which I've never heard about, which apparently neurons have, and ATP can bind to it. And it's one of those
Gproteincoupled receptors.
Exactly.
Yeah, so there's two different types of receptors. The Gproteincoupled ones are basically just like more complicated. Like you bind to the receptor and then it sets off this whole like Rube Goldberg machine style of different interactions in the cell that can lead to like just about anything potentially. Versus
including changes in gene expression or inhibition of the cell or things
excitation of the cell too.
Anything, versus the more simple receptors where they just kind of open an ion channel and let ions in. So it seems like the glial cells work in this like more complicated way. You're saying that like neurons have ATP receptors that do this more complicated type of effect on the cell, which probably means that it's harder to see what the effect is because it can happen over a longer time scale and not be as obvious.
there's slower effects because they're through these G-protein coupled receptors.
Yeah, so I mean, I think a lot of these points are highlighting, well, one, that glia could be doing very interesting and relevant things for sort of circuit computation. Two, that it is hard to study them in part because, maybe people have known about it for a shorter period of time, and it might be harder to see what's going on in a rapid way, which is related to the third point, which is that they're on a slower time scale. Mm-hmm. The slower time scale means that it's also harder to see the direct consequences of what's going on. So both the activity of the glia seems to occur on a slower time scale, but also the consequences of it in downstream neurons, if it's mostly doing things like activating G-protein coupled receptors, the effects of glial stimulation or the effects of glial activity could be delayed or slower in terms of taking effect.
Yeah, and also, the tools we've been developing have been to study neurons. And so that's what in one of these reviews, they say, we need better tools to observe and manipulate glial cells, which is where the optogenetics and the more modern tools come in. It happens to be the case that, we've developed fluorescent indicators to let us know when calcium is being released inside a cell, because that helps us monitor neural activity, because neurons release calcium when they're firing. But now we could use that to study glial cells. So it's kind of just a matter of people weren't, searching to build the tools to study glial cells. They were building tools to study neurons that we now can use to study glial cells. Also, it was interesting to me. They mentioned briefly the history in one of these reviews. In the 2008, they referred to a paper that goes through the history of glia, and I kind of skimmed it. And it was surprising to me almost that they were viewed as connective tissue and supportive cells, right from the start, it seems. There were people who didn't even believe that the brain had any connective tissue in it, and there was somebody who was like, no, it must. And they found glial cells. And it's kind of surprising to me if it is the case that they are 1-to-1 in the human brain, or even 10-to-1. I guess I would have thought that somebody at some point would have thought that they were the main thing. But it seems like being electrically excitable was very important from the start of the history of neuroscience, and the fact that glial cells didn't respond much to electrical probing kind of put them out of the running.
Or at least they didn't exhibit short-term electrical responses in response to electrical stimulation.
Yes, of the magnitude that neurons show.
Yeah. But I think a related point to that, it does seem like if someone in the 19th century took a kind of half-blind stab into the brain and took a guess that glia were glue and left it at that, it would be surprising if that were correct. Just in terms of the history of science.
Yeah, no. I was surprised that, well, okay, you're saying it's not correct.
Well,
that it's not just glue.
Yeah, no, clearly, it's already clear from the evidence that glia clearly are more active than that. Yeah. And I don't know. I don't want to we're talking about some of what we know, which is not that much. Ultimately, we're not going to get that far in this discussion because not that much is known. But if in fact people were right for over 100 years or whatever, that glia don't play a critical role, I don't know. That strikes me as like a lucky coincidence, given how little was known when they made that claim.
No, of course. Yeah. And I think we already have enough evidence to know that they're not just some kind of glue. They have more active roles.
Yeah. It seems like it. I guess, yeah.
What I'm saying is I'm surprised people weren't more wrong. I thought that people would think that glia were everything just because there's so many of them.
Sure, okay.
I had less faith in people of the past.
If you think about the early neuroscientists, once they discovered that there was electrical signals in the brain, I could imagine oh, that's not electrical, so then that must be the matrix around it. That's, that's not important. So maybe there was just these strong biases to it, if it doesn't have electrical fast signals, then it must not be doing anything.
Yeah, no, that definitely seems like what happened.
And then glial cells are definitely the underdog of neuroscience, meaning people that are studying the brain, very few people were thinking of studying the cell type that didn't seem to be the main cell type, which actually I think is just wrong. based on the estimates of like 50% of the cells are glial cells. So I think glial are becoming its own field.
I would be interested to hear your perspective as someone who does experiments. I know that like glial cells aren't studied so much by most labs, but do they even come up as you're like talking about, like, are there practical considerations that come up when doing an experiment that involve glial cells?
The only practical consideration that I have to face is gliolysis happening surrounding your electrode over time.
Yeah.
Can you explain that?
So, because glial cells are protecting the neurons around it, when there's any type of lesion, like you imagine if you're implanting an electrode in the brain, you're creating a hole in the brain, you're creating a lesion. So the glial cells are going to try to protect the neurons that are around it from this lesion that has happened. So they're going to create a little crust over time.
You can think of this as like a kind of immune response or like an inflammatory response.
Scar tissue.
It's like scar. Okay,
it's scar tissue, essentially scar tissue. But surprisingly, we don't talk about glial cells in like lab meeting and being like, oh, I wonder if glial cells have anything to do with this. Nobody talks about it. So it hasn't really crossed that threshold.
Or a related point is when, for example, doing imaging, based on the approaches that people use, I guess neurons are preferentially stained or fluoresce, whereas glia don't. So they're essentially transparent for the purposes of the imaging approaches that people use.
Yeah, people design their imaging techniques so that they only make neurons glow. They only make neurons visible. And the same thing with if you're dropping an electrode in, if glial cells aren't having a strong electrical signal, if that's not how they communicate, then it won't be something that you have to talk about. Like, oh, I'm worried that this is a glial cell and not a neuron. Like, you won't see the signal from them at all.
Right.
I would say as a computational person, glia come up sarcastically more than they come up with any sincerity. It's usually in the terms of like, people are complaining about how hard it is to understand the brain and neurons are so complicated, and then they jokingly say like, yeah, and it'll turn out it was just glia all along or something like that. It's like a throwaway joke to point to how little we're confident about our understanding of the brain. So yeah, really not talked about with much sincerity, save for like the handful of people who explicitly study them. It's not really integrated into our understanding of the brain.
I like that joke. I'm going to start using it.
But yeah, so I keep wanting to say neurons. That's demonstrative of the problem right there. Glial cells, according to these reviews, can impact the strength of synapses and other elements of how neurons communicate with each other. And the fact that they work on slow timescales, I mean, we talk about that as being, a sign that they're not neuron-like and potentially that they're not, really used in communication. But when we see slow timescales in neural activity, when you can find it, we call it memory. So you could argue that the slow timescales of glial cells are a form of memory. They can hold on to kind of a trace of the overall neural activity that's occurred in an area and use that information to guide synaptic plasticity, which is kind of the story that emerges from these reviews.
Yeah, which is actually really cool and interesting and like mind-blowing for me as a neuroscientist to think about, glial cells are part of, are important for making memories. It's like, whoa.
Yeah, so the things that they say about the role of glial cells, when they stimulated the calcium waves in the glial cells and then measured the synaptic strength between two neurons, it seemed like the synaptic strength went down, like the strength of how much one cell influenced another decreased.
So it decreased synaptic transmission.
Yeah, so they did these experiments in the neuromuscular junction, which is in the peripheral nervous system, which admittedly is different from the central nervous system. But the experiments that they did said that if there's an increase of synaptic activity between the neuron that connects to the muscle, where it's kind of controlling the muscle, if that's increased, then that leads to this like calcium activity in the glial cells, and that leads to a decrease in the strength of that synapse. So like neuroscientists are used to seeing adaptation in synapses, meaning like the strength of the synapse goes down if you use it a lot. It's just like a known thing that if a synapse is activated a lot, the strength of it might decrease. But I don't think they usually think about it as being mediated by glial cells. So this is saying that adaptation at that synapse happens because of glial calcium signaling and what the glial cells release in that synapse.
That's really cool. Even though the data is not here, it wouldn't be hard to believe that that's the case also in the brain. That there's some sort of modulation, not necessarily these very same, but that there is some sort of modulation of synaptic transmission, considering the glutamate release or the ATP, the fact that ATP can modulate neuronal activity or bind to receptors on neurons.
Yeah, there's just like so many pathways when you're really looking at the full picture. Like there's so many different neurotransmitters and different effects that neurotransmitters can have on a synapse. It's like almost anything is possible when all of a sudden glial cells are sending signals. They also talk about a study in the hippocampus. So it seems like most of these studies are in either cultures, like grown neurons in a dish, or in slices from the retina or from the hippocampus. I'm not sure why those were the two places. I mean, the retina, I guess, is like a nice, clean brain area because it's just an input layer, and so maybe it's easier to kind of separate it from the brain and treat it as its own circuit. Hippocampus, I think they study this in just because synaptic plasticity is really big in hippocampus, and so if glial cells are involved in synaptic plasticity, like that's a place to look for it. Like the connections in hippocampus are changing all the time.
Yeah, and then there's existing protocols to induce LTP. So if you wanted to make a claim, it definitely makes sense to study hippocampus. If you wanted to make a claim about plasticity, hippocampus is definitely a good brain region to study.
Yeah, and so the thing that they talk about in hippocampus, so first of all, they said that 50% of the synapses in hippocampus have an astrocyte surrounding them, and they have this picture, and it's like, if you've seen a picture of a synapse, it's like, it's the normal picture of a synapse. There's like the end of one cell and then a little bit of a gap and then the start of the other cell. And that's like where they communicate with each other. But then they just have this like third thing that kind of like is just wrapped around the thing, the two
it completely
neurons.
Envelops the entire synapse.
Yeah, it's like, and it's just crazy to me that exists like on the synapse level. I kind of thought of glial cells as like being more like diffusely placed, not that they're like right up there in the synapse, like wrapping around it in like a synapse specific way. And it's also just weird because like I've seen a drawing of a synapse a million times and no one ever puts the glial cell in there. Yeah. So it's like a third cell that's forming the synapse, at least in hippocampus, and 57% of the synapses. So it's just kind of crazy to see it, and like when you see it in there, you're like, this must be doing something.
Yeah, and then you also wonder what makes those 57% of synapses that have glial cells around it special. Like,
oh yeah,
can they have LTP easier or harder, or what? What is the reason? How does it differentiate them?
Yeah, so they say like going back to like the classical picture of glial cells, part of the role that they play is to do reuptake of neurotransmitters. So when you release the neurotransmitter into the synapse, you don't want it lingering around forever, and the neurons do some of their own reuptake, but I guess glial cells also take in those neurotransmitters just so that they're not lingering there. You don't want them to like keep activating the cell beyond the point where they were supposed to. So that's one thing that it does. But then this study that they talk about, they said that if you cause glial calcium signals, it leads to an increase in the strength of the inhibition. That, like, if an inhibitory neuron is connecting to another neuron, calcium signals in astrocytes is correlated with that inhibition getting stronger. And then they do an experiment where they actually shut down like the synaptic activity of the two neurons so that you know that that change in strength isn't just because the neurons are doing something at the same time. They say like, no, it's like really the glial calcium signal somehow leads to increasing the inhibitory strength of the synapse, even when the neurons aren't doing anything, which is like very relevant if it's true.
Yeah, so in a more recent study, they mentioned GABA B receptors are activated by astrocytes because of calcium influx. So it might be mediated through GABA receptors, the B version instead of the A. So the B receptor of GABA is a G coupled receptor. I said it right now. Instead of a channel, which is GABA receptor A. So when we think of fast inhibition, is through the channel, which lets chloride in. So it seems like it's very common for the transmitters, ATP or glutamate, that are being released by astrocytes. They're activating Gproteincoupled receptors.
Yeah, exactly. But yeah, so like, what does this mean? Like, what could be happening? The fact that the glial cells work on this like slower time scale, and also they like pool across a bunch of different synapses. Like one glial cell can have contact with many different neurons and synapses. So it seems like why would you want this? Are they like taking in some readout of overall activity in a brain area and then saying like if the activity is high, then you should be making your synapses more plastic, or like letting them move in a certain direction?
You could imagine that they're doing some, I'm speculating, they could be doing things related to like homeostasis across many synapses, which would be related to your point of getting some sense of the overall activity levels in order to somehow modulate it. So that would, you could view this as a computational role, and it's clearly a computational role, but it's also a sort of supportive computational role in the sense that it's not engaged in direct computation, but maybe some sort of kind of indirect diffuse normalization or something like this. So, people view normalization as a tremendously important computational role, so I'm not trying to necessarily separate it. The other thing that happens, and there's been some literature on this, is a connection between glial activity and bold signal. So for this kind of diffuse slow timescale activity could have something to do with blood vessel dilation or contraction or constriction. There have been some sparse studies indicating that glial activity is related to or correlated with the bold signal, right? So
yeah, no, it's they talk about how this glial activity is correlated with blood flow, which is what the bold signal is measuring in fMRI. But also, like, glial cells can release vasoconstrictors and dilators. Like, they have the ability to release things that control the blood flow. So there's, like, a very clear mechanism for how they relate to blood flow.
Yeah.
So I think we didn't mention that astrocytes are actually, some astrocytes, not all of them, but are physically touching the blood vessels. They're part of the vascular unit. So you've got an astrocyte that could be, as Grace was describing, like wrapping around one synapse, but that same astrocyte could be also wrapping around the blood vessel.
So again, the cynical view, right, is that glia are monitoring the activity of larger numbers of neurons in order to sort of
provide oxygen.
Well, yeah, vacuum up excess neurotransmitters, so to keep everything clean, and provide oxygen and nutrients to cells that are more or less active in proportion to their activity by regulating blood flow. So that's like an important supportive role again, and it requires more computation than maybe people initially thought, right? The fact that blood flow is dynamically routed to different cells based on their activity levels implies that there is not just responsive coding, but as people have shown with respect to the bold signal, some of it's a bit anticipatory, right? So like if neurons are about to fire, the blood vessels that go to them often in sort of they'll proactively start siphoning blood to areas that will become active. That's at a coarser level of resolution than single neurons, right? But the sense of this is that there's a whole computation related to routing blood and maybe even doing predictive modeling of where blood will need to be routed based on anticipation, anticipating what areas will be more or less active based on activity. So that's deeply computational, right? There's definitely computations going on, but it's indirect in the sense that it's a kind of meta computation that's like computation about kind of gross computational properties of the neural circuitry.
I like that model a lot, but I don't see how then neurons having receptors that are activated by the things that glial cells release makes sense. Because in this model that you're describing, glial cells should be able to monitor, meaning they have receptors that respond to neurotransmitters. That's great. And then they can modulate the blood vessels. Perfect. That does happen. But why will glial cells need to release neurotransmitters, essentially, that are then going to be binding on the neurons? Like that.
Yeah,
why would you need that?
The bidirectional aspect of it does make it seem like, the glial cells are trying to tell the neurons something, or at least the neurons are responding to what the glial cells are doing. Whereas a purely supportive role, you would expect just the glial cells to respond to what the neurons are doing.
Yeah, no, I mean, I completely agree. I mean, it can't be as simple as what I described. I mean, I think
I liked your explanation at this time.
There are extra things that could go on. I mean, we already have acknowledged, I think, that there's likely a role for normalization. Right, so if you're simply monitoring activity of neurons across a larger spatial area, and we've said that certain kinds of glial modulation increase inhibition locally. I mean, the kinds of activity, the bidirectional signal, the response signal from glia back to neurons could partly be related to normalization or homeostatic kinds of mechanisms.
Okay.
Which are not totally inconsistent with some generalized view.
So another area where the glial communication to the neurons comes up is in development. So we've kind of been talking about the way a synapse can change, assuming that the animal is kind of in a mature adult state with its formed synapse and stuff. But also glial cells are really important for the development of synapses between neurons in the developing brain. So they talk about, in the 2008 review, a study that tried to grow retinal cells in a dish and showed that if you don't put in the astrocytes, the cells can grow and they can become they can become excitable and they seem otherwise normal, but they don't actually have effective synaptic communication. They won't talk to each other through synapses. And so if you put in the astrocytes, though, then their synapses form correctly. So that suggests that in the developing brain, the formation of synapses is dependent on astrocytes. So the protein that the glial cells release that's important for this is called thrombospondin. And so they say that this protein, you can measure levels of it, and it's high during development, which makes sense. It's also high after injury. So presumably synapses need to reform after an injury. And they said that it's high just in the hippocampus normally. So that's in line with the hippocampus being part of kind of quick memory formation, that they need high levels of synaptic genesis and synaptic reformation.
But again, chiming in as the cynic, I mean, it could be the case, right, that often underrated in the development of the nervous system, or even the sort of functioning of the nervous system, is the sort of structural elements. And for a synapse to form, you could imagine that there's some amount of extracellular structuring that takes place. And it's possible that if you remove glia, which we know to be responsible for sort of maintaining extracellular matrices, the structure of the sort of extracellular area that synapses have trouble forming, not because of some sort of computational role that they're playing during their development, but due to sort of structural reasons.
But these things get blended. I mean,
those are related. Yeah. I'm not how does the glia know where it should be adding the structural elements that it's building? It's responsive to neurons in order to do that. So I'm not trying to segregate these completely, but the idea that the glia are not directly involved in the computation of a healthy system could still be maintained, if you wish, right, by claiming that while the glia are responsive and it part of this developing circuit, the role they play in that developing circuit is not a computational one directly. I mean, what computation means is obviously...
Yeah, so what computation means is complicated, but I would say there is another element to this that I would say argues for more of an interesting role in this protein that leads to synaptic formation, and that is that the glial cells will release more of this protein when ATP is around, and neurons can release ATP. So it could be that the neural activity is telling the glial cells, release this protein because I want more synapses. And that has the flavor of a more computational role because it's actually based on what the neurons are doing and whatever inputs they're getting and their activity and all of that.
Yeah, so I think a lot of this gets at what people mean when they say computational. I think it's clear that the sort of extremely naive notion of glia simply as there and providing nothing but support is kind of naive because they're clearly adaptive and responsive to stimulation by neurons, and they also in turn signal back. But then the question, the more nuanced version of the question becomes, are glia involved in computations as like the kind of thing involved
action selection, like,
coordinating movement, action selection, forming memories, learning, or they're clearly involved in learning indirectly if they're required for the supporting of the synapse, granted, and they're clearly involved in routing blood flow, perhaps, as we've been talking about, and that seems like a kind of movement-oriented thing. So, I mean, from a very generalized sense, like clearly they're involved in a kind of reactive or computational way in certain functions. So, but then the question becomes, and it's this inching standard of like, are they really involved in like what people think of as core computations of the brain?
Well, I think there's computation used in the sense of like on the fly, moment by moment, how do you process your inputs? But the study of learning and synaptic plasticity is a huge part of neuroscience, and it's its own different type of computation that happens on this different time scale and relates to the computations that are done, normally, like what you've learned relates to how you process information, but learning is like a little bit different. So yes, if it's involved in learning, particularly to me, it's relevant that there's some sort of feedback between the neurons talk to the glia and the glia talk to the neurons. If it's involved in synaptic plasticity and learning in that way, then it seems pretty relevant, I would say, as, there's reason to talk about it in a computational role. Kind of like the decisions the glia make will impact what is learned, and that seems relevant.
The decisions that glia make will impact, say that sentence again, Grace.
I think the point is just like, if glia are responsive to what neurons are doing, and their participation is required for for effective learning. So like if they're required for supporting or building new synapses, then glia are just integrally part of the learning process. So to like view synaptic plasticity as something that happens and have discussions about synaptic plasticity without considering the sort of clear role that glia play in that, let's even say computationally, insofar as they're part of building new synapses, then yeah, I think that
it's kind of like, for it to be really supportive, it would seem like if the glia behaved differently, it wouldn't really matter in the long run. Like they're just, it's like when you say like support staff of an organization, it's like there's the people making the decisions and then there's the people who are like providing them lunch. And if they provided a different lunch, it wouldn't really matter.
Yeah, but I think the statement would be more like, if you have an organization or you have a city, and the people who fill the grocery stores stopped filling the grocery stores, the behavior in that city would start looking different. Like people would start looting, or people would starve, right? I think that I guess I'm coming in with the stronger version of, they could still be playing a primarily supportive role, even if they have to be responsive to what neurons are doing. And even if neural activity changes when they're not there, due to like health or blood flow or lack of vacuuming up neurotransmitter. I mean, I don't want to be like the voice of this naysayer case, because it's not like I have reasons to side with this. It's tempting, I think, in an era when people have gotten fairly far in neuroscience without so much study of glia. In the early 2010s. Around the time that both of these papers were written, so like between 2008, 2011, or whatever.
2001.
2001.
Yeah, it was like mid-90s to early 2000s that this view of the glial as being more important than just supportive was.
Yeah, people wanted to say something like, glia are more important than you expected them to be.
Which is, yes, a relative statement.
Yeah.
With a medium level of impact. It doesn't mean something absolute about the brain, I understand.
So, Josh, is there an experiment that you have in mind? Like, well, if they, if someone, it's a hypothetical experiment. If someone manipulated, the glial cells, is there anything that would convince you otherwise? That it's not purely supportive and that it's part of the integral computations?
So, I think we have to be clear. This is a sense people have as a field. It's like a perspective, kind of, that is broader than a single experiment. But I think, so the question would be, can you show somehow that glia are like integrally important for like something that we consider like very, very computational? And as Grace has already been kind of highlighting, if learning doesn't happen correctly when glia are absent, just sort of straightforwardly, you could say, oh, well, glia are then definitely important for learning. They're necessary for learning. But the question is more, what is the role they play in learning? It's not, are they necessary for learning? Because that's like a fairly weak statement. The question is, when learning happens, when a new synapse forms, are glia involved in deciding like something about that synapse, like where it forms or how strong it is? Or are glia merely responsive to primarily neural signals, and it sort of responds or reacts and says, okay, I will envelop this new synapse and provide it with the support that it needs. And that requires responding to it. Right? You couldn't have that new synapse without the glia. But the question was, did the glia have any sort of control over or like a role in the decision about the formation of that new synapse, or did it simply respond to it? And so this is not like, I'm not proposing like a single experiment. That's a perspective. That's a model of what's going on with glia that may or may not be right. I think this would require like figuring out that glia are somehow more relevant than that. So like maybe you could show through a series, like it would really be like a research direction, that something about the activity of glia determines which neurons are likely to spawn new synapses or something like this, right? It would require like a more concerted change in the way people conceive of what glia do relative to neurons, I think.
Yeah, it feels like the neurons are in some way making decisions and the glia are helping them carry out their decisions in terms of like forming synapses or whatever. Except for some of these things that say that when there's a glial calcium wave, the synapse can decrease or can change in some way. That feels a little bit more like proper influence, although it's, again, probably not very specific because these signals from the glial cells are kind of spread out over more than one synapse.
Implying maybe like a very diffuse normalization or something
Sure.
related to routing blood flow to a general brain area, right? Or something like that.
Yeah. There's another study that I think is cool and probably doesn't change anything that you're saying, but I still want to talk about it. They were saying that this protein that causes the new synapse release, as I said, is released and like in high levels during development. And so during development in certain systems of the brain, there's something called a critical period, which means that types of learning in that area can only happen during that time. And if you kind of don't get the right sensory inputs, say for your visual system, then like it just won't develop right, and then it's stuck that way. So once the critical period is over, like just certain things cannot be changed about the system. But if you take astrocytes from an immature animal whose critical period is still going on and put them into the cell, into the brain of an animal that's mature, you can reopen the critical period and get types of changes in connectivity and responses that usually only happen during development.
Wow.
So again, that's still probably like the neurons, have some activity that tell them that they want to form synapses, but the glial support isn't there, and you can put the glial support there to like let them form those synapses. But it is very interesting because critical periods in neuroscience are like considered like pretty hard limits. Like the critical period is closed, this type of learning is over. So anything that can reopen them is like a pretty strong thing.
Yeah, and from therapeutic perspective as well for degenerative diseases, maybe that sounds really promising.
Yeah, true. Let's talk about some diseases. Most of the diseases related to glial cells are surrounding myelination, it seems. So, if the myelin around an axon fades away, then the axon is not insulated from the electrical signals that are happening around it, and it can't properly send its signal, and that leads to problems. And multiple sclerosis is one of the diseases that is a problem of demyelination. It's an attack from the immune system on the myelin, and the cells that produce the myelin, the oligodendrocytes and the Schwann cells, I think they said they try to respond and rebuild, but they're limited in their ability to rebuild after the immune system has attacked them. Another surprising disease that they said was related to myelination is depression. According to this paper, there's actually a very strong effect where people with severe depression have lower levels of myelination, which was surprising to me.
Yeah, I tried looking, I was talking with my glial expert friend, and she said that there's not just one paper, that there's now more and more evidence linking glial activity or glial changes with depression. So of course it's hard to know what came first, like the egg or the chicken, right? Like, is it that there's other problems that inflame, that activate? We haven't talked about this, but when, because glial cells can have immune responses, if you have cytokines, which are molecules from the immune system that signal a problem, if you have cytokine present, you can activate the glial cells and then they become reactive. So it seems like in depression, there could be decreased myelination, but you can also have microglia that are reactive. So it's hard to know what came first.
And just to sort of phrase this in a slightly more colloquial way, right? There's a sense generally that things like stress or things like this weaken the immune response, right? There's sort of a lot to back this up.
Right.
And so, like, a claim might be that if you are depressed for a very long period of time, either you're sort of experiencing a stress response that somehow taxes things, and maybe a consequence of this is that the immune system, for example, attacks myelin in certain parts of the brain, and this is like a signature or a symptom of depression. Alternatively, it could be the case, like with many autoimmune diseases, that there's an autoimmune response which is primary in some sense, maybe due to some sort of genetic anomaly that causes the destruction of myelin in certain brain areas, and then that results in depression. Mm-hmm. So,
well, they also say that the oligodendrocytes that produce the myelin also release something that's involved in the synthesis of serotonin, which is a neurotransmitter that people associate with depression. Like, depressed people don't have enough serotonin. So I feel like that puts in a connection as well. But it could also be kind of like a red herring that they just happen to also release this thing because they release a bunch of things.
It's also, I'm not sure if it's mentioned in the 2008 paper, but there's a similar link of between Alzheimer's and neuroinflammation being high.
So I guess the immune system connection is mostly through the microglia, because they release these cytokines and perhaps are responsive to them as well that are part of the immune system. So yeah, that kind of opens up the door for the brain to be influenced by the immune system in like a million different ways. Which just makes things so complicated, because I think people talk about the immune system as like kind of on par for complexity and difficulty to understand as the brain is. So we're just like bringing in a whole other really complex system that can influence this really complex system.
Totally. And then it means that you can have glial cells responding to things that are happening in the periphery through the immune system, and then potentially influencing local neuroactivity, or at least the extracellular space surrounding neurons.
Yeah, exactly. And the 2008 paper, they talk about targeting glial cells for developing drugs, like trying to interact with the glial cells, because there's been a bunch of attempts to, fix things like Alzheimer's or depression with cells that are supposed to be targeting neurons or neural transmission or something like that. But like in neuroscience more generally, it seems like medical neuroscience doesn't pay a lot of attention to glial cells. And so I just liked this quote from the paper. It said, "Glia already know how to save neurons, whereas neuroscientists still have no clue." So they're saying, help the neurons or help the glial cells, and the glial cells will help the neurons. And so that's a possible route for therapeutic developments.
Yeah, and that quote was pretty representative of the writing style of this author. So if you're looking for a fairly
fun,
let's say a fun or playful, a playfully written read, then this is the article for you.
Do we have any final thoughts?
I would like to hear what Josh's definition of computation is. But before that, I guess my takeaway after this, even if I remain skeptical to like, how much of a driving force are the glial cells for the very important like functions or for driving behavior. I care about behavior, so for driving behavior. I'm not sure, but I could imagine that if people study glial cells, especially astrocytes, which seems to be like the most common one, we might get a better understanding of neurotransmission, and that might help us. So I think that more people need to study glial cells, because given that there's so many of those cells in the brain, I could imagine that they're doing a lot that we're not aware of, even in a supportive role. But maybe having that knowledge might help us understand neurons.
I'm in favor of that message. There's enough neuroscientists trying to do stuff. Some of them could be more focused on glial.
Or maybe the people that are interested in the immune component can study glial cells. If maybe immunologists can gravitate towards neuroscience through glial cells. Like we could recruit extra people.
Yeah, take the funding from immunology and use it to understand the brain. I'm also in favor of that.
I mean, getting to the point, you were asking, like, what do I think the definition of computation is? I don't want to provide like a comprehensive definition. I mean, I would say we've talked about a few candidate roles for what are even supportive computations, like routing blood or predicting activity in order to anticipate energy demands by brain region and then routing blood in a sort of proactive manner, or responding to neurons and helping to build sort of new synapses or things like that. Those are definitely computational functions, right? It involves doing things in response to stimuli. I think where I would say maybe it still like fits a loosened version of the classical narrative or the conventional narrative of glia is that they are like supportive computations, perhaps. I'm not wedded to that view, but like if you believe that it's only those kinds of things, then you could argue that they're like supportive computations in the sense that the neurons are doing the sort of interesting things. The neurons are involved in working memory. The neurons are involved in perception. The neurons are involved in control of the body, so motor coordination, action selection, planning. Maybe glia are only involved in these sort of secondary roles, not these sort of core computations that we say are fundamental to behavior. It's not about computational versus non-computational in a strict sense. It's more about types of computations being performed. Given everything that has been learned so far, that division of labor doesn't strike me as totally insane.
Considering the connection between the immune system and glial cells, I think that the neuroscientists that are trying to study any disease state, any animal model of any disorder or disease, are definitely ignoring glial cells. Like, that's a place where I wouldn't be surprised if a lot of phenotypes of the disease or disorder, even including some behavioral problems, come from a change in glial cells. Just because they respond to cytokines.
So you're saying, like, for example, it might be the case that you're in some state where things get unhealthy and then, I don't know, glial cells inflame or damage parts of the brain.
The neurons around it, right,
yeah.
Exactly. Like damaging the synapses, for example, like pruning the synapses that weren't supposed to be pruned because immune response. Wait, what's your takeaway, Grace? You didn't say anything.
Yeah, I guess reading this, I saw what I was expecting to see, which is that they do have very important roles that are aligned with the classical picture of them in terms of, myelin and all these other things. But yeah, I guess some of the fine details of their influence on synapses could quite accurately be construed as a computational role that would be relevant to people who study synaptic plasticity, especially given that there's just a lot of stuff about synaptic plasticity that we don't understand. It's like, well, if there's this other factor that could help you make sense of your data, then take a look at it.
That's a good point. So people studying synaptic plasticity, learning, and people studying disease, they're the ones that should definitely be studying glial cells as well.
All right. Thanks, you guys. Thanks, Nancy.
Thanks for having me, even when I barely know much about glial cells.
Join the club. All right, till next time.
Bye.
Hey, if you're still listening to this, you must really like us. So how about you go to iTunes or Stitcher and rate the podcast? Give us some feedback. You can also go to our website, unsupervisedthinkingpodcast.blogspot.com. You can comment on different episodes, or you could give us ideas for new topics you want to hear about. We would love to hear from you. Thanks.